Et me revoilà pour le numéro 2 de Rethinking Cancer, conférence de Jean-Jacques Trochon. Et là, c'est William Li qui nous parle. William Li, un grand monsieur que je n'ai pas le plaisir de connaître, mais que je vais connaître bientôt grâce à Jean-Jacques.
I thought as the first speaker, and as a vascular biologist and a medical doctor, my area is internal medicine, so I think about the whole body, but have spent many years looking at cancer. I thought I would really focus on rationale in terms of validating the idea of dietary cancer control, which is along the theme of developing evidence. And I'm going to talk about sort of the big picture of diet and cancer, and I will bring it to my own experience in terms of how evidence is currently being developed. And when I say currently being, what I'm saying is that we are still very much learning how to actually develop this evidence, and so as it accumulates, we can put it together, integrate evidence together as we will during this day, and hopefully have more scientific and clinical collaborations that will allow the field to stand on its own and not be viewed as sort of a side field that is not mainstream, but in fact to bring it into the mainstream. As Bernard has alluded to, my work in angiogenesis, which led to our introduction many years ago, this was angiogenesis itself was a side field. It really took a focused scientific approach married to a clinical approach in order to be able to bring validation to it. So with that, I'll start with this infographic that shows different causes of death in the world. And of course, the odds of dying from any cause is 100%. We will all go for some reason. And when you look at many different types of reasons, like drinking too much alcohol, or drowning in the swimming pool, or perhaps having an automobile accident, which is tragic and common, you can see that cancer, in fact, trumps many of those other causes, and in fact is only second to cardiovascular disease as a common cause of death among many different ways that we will all go. This is perhaps why cancer research is an excellent focal point for examining new fields, such as the impact of diet and health. And what I'm looking at the program and imagining is that many of the scientific concepts and even clinical implications that we'll discuss today not only apply to cancer, but may apply to other diseases of life, including chronic diseases, but potentially, perhaps even more importantly, give us some insights into how to remain healthy and how to protect our health early on. The other sort of importance of cancer, of course, is the scale on a global level. If you look at numbers from 2010 to projected to 2030, this is the new data from the AACR showing that the growth from 13 million cancer cases to 21 million, this doubling, is going to occur if we don't actually try to prevent the disease. And smoking cessation is one of the important interventions, but there are many others, and people approximate perhaps up to a third of cancers may be associated with diet, and oftentimes the association is what is the negative element in our diet that we should avoid to avoid cancer. But what we're here to talk about today is, are there other ways to view diet, including fasting or metabolic control, or the intake, consumption of certain dietary bioactives, which I'll talk about. The other issue is economic. Cancer is a very costly disease. It's costly in direct costs of treatment. It's also very costly in terms of indirect costs of loss of income and productivity for the countries. You can see that if you project from the $290 billion of cancer costs from 2010 to almost $50 billion in 2030, this cost will exceed the current GDP of these 5 countries, Austria, Norway, Denmark, South Africa, and Ireland. So it's quite stunning to think about the cost. Now, much of the cost is involved with treatment, and the new treatments for cancer, which I will also mention briefly, are very exciting but also very expensive. And so the other implication of diet is that there's maybe an opportunity to help not only save costs through disease prevention or cancer prevention, but also potentially an adjunctive treatment using our diet that is largely ignored by the oncology community, really because of lack of data and lack of education, to be able to achieve clinical goals that we would like to have. So the topic of validation is not one that's only for diet. In fact, from the very beginning, in the history of oncology, surgery had to be validated, chemotherapy had to be validated, and radiation had to be validated. And these are all the mainstays for cancer treatment. And while they often fall short from what we would like to achieve with patients, they can also be quite effective in some cases. And in many cases, they allow the patients to live longer from the time of diagnosis and represent the best of practice that exists in many places around the world. Now, this has changed over the last 15 years, maybe almost 20 years now. Targeted therapies worked through very difficult beginnings with just discovering what are some targets on the cancer cell, in the microenvironment of the cancer, in the matrix, in the vasculature, and now in the immune system, which has led in kidney cancer as a beginning, but many other cancers now, lung cancer, endometrial cancer, melanoma, to give us an understanding of how our own body's defenses through the immune system may wind up becoming much more powerful than we ever imagined, number one, and number two, perhaps more durable than our other treatments at hand, like chemotherapy. And so I just want to show you some of this. A human face always helps to make the case. This is my colleague Stephanie Joho. She's 28 years old. Her mother died of Lynch syndrome, which is an inherited genetic time bomb for multiple malignancies. And she was in college when she had bleeding in her stool and was went to the emergency room and diagnosed ultimately with stage 3 colorectal cancer. And at the time, she went to very good oncologists in New York City and sought treatment, which was involving surgery and chemotherapy. They considered some radiation, and her cancer continued to spread. And she was actually given a palliative approach with just going home with pain management. Her cancer was essentially unstoppable using the traditional, conventional means. And her younger sister, as so often happens today, works in the media and was very media savvy, went on the internet and found a clinical trial at Johns Hopkins University on immunotherapy. And by looking at the molecular profile of her colon cancer, they found a marker which suggested that she would respond to an immune therapy, a checkpoint inhibitor, and indeed, within a few months of treatment, in fact, all of her tumors disappeared. And so this is what she looks like today. This has now led to, among other work, an FDA approval on the molecular signature for microsatellite instability. She's been in the media, sort of like JJ has, as a champion of trying to move progress forward from the patient perspective. There's also a companion to these new treatments, which is to do molecular profiling in real time. I'm going to come back to this at the very end, but you can look at the entire human genome in the tumor, compare it to normal leukocytes and compare that genome, look at the transcriptome, look at the proteome, use artificial intelligence to compare, look at what the targets are, and then select treatments in a precision fashion. And there's also now liquid biopsies allowing you to use blood to look for circulating tumor DNA and RNA. We're at the very beginning of this, but I'm already involved with some of this type of work. Now, angiogenesis, which is my area, describes how the body grows blood vessels, and tumors are able to hijack that process of growing blood vessels in order to be able to feed themselves with oxygen, nutrients, growth factors, and other survival factors. And so within the environment of the cancer, blood vessels become one of the actors that actually help to enable the cancer to grow from a small, essentially harmless mass, as you can see on the left, because we all are forming cancers in our bodies continuously, even now. And then when they are able to grow a vasculature, they can actually trigger explosive exponential growth. In the laboratory, once blood vessels touch a tumor, the cancer can grow in volume 16,000 times in just 2 weeks. So the idea is, can we actually cut off the blood supply to tumors? This is something that was pioneered by my mentor, Judith Oakman, and then subsequently more than 1,000, now 10,000 researchers and clinicians have entered the field. And it's led to a number of anti-angiogenic agents that have been FDA approved starting in 2004, as you can see on the top, first for colorectal cancer and then for other types of cancers. And now there's 20 different human anti-angiogenic cancer treatments that belong to targeted therapy. There's even one in the middle, which is in 2009, toceranib, which went from humans to animals. The veterinary world actually borrowed from the tyrosine kinase inhibitors for angiogenesis for canine mast cell tumors. And so this is where we can learn from our animal companions, not only in the laboratory as mice, but also to try to give back to the animal world. And this field has been able to cross-fertilize that way. So to move to our topic, what about validating dietary control of cancer? It's not a new idea. In fact, if you look in other cultures, Asia, many countries in Europe, in Germany, and including in France, the tradition of diet in health and diet in cancer goes back a long time. But it has, until recently, not had the scientific foundation and the integration that I'm going to show you now and we're going to hear about today. So the first thing as a scientist, as a vascular biologist, I ask in terms of validation are some basic questions. So first, does it work? How does it work? How do we apply it if we want to actually use it for treatment, whether it's in a laboratory or in the clinic? And then how do we monitor the results? And I think that if we can begin to ask these questions in responsible and rigorous ways, then we can establish the basis for really using and diet methods as a mainstream, as a companion in everything else that we have for cancer. So it's challenging to validate dietary cancer control. And you know, many of us who work in and have experience in the biopharmaceutical area, including here at the Institut Gustave Roussy, is we always think about developing evidence as randomized, double-blind, placebo-controlled trials. Very difficult to do that with food. There are some small studies that attempt that, but then they are small studies. It's a very difficult in a large population way to actually tell people only to eat tomatoes or potatoes or certain vegetables for long periods of time that we may need in order to see these outcomes. And so I think finding this level 1 evidence, which has become one of the gold standards of medicine, is something that we all need to aim for, but recognize how difficult it is to do here. Now, the other way to do this, besides a pharmaceutical style trial, is really to look at compiling the different types of evidence that exists at the genomic level, molecular level, in vitro or cellular level, looking at animal data, clinical studies, which tend to be small, 10 to 100, well-designed, controlled studies have been done in foods. And then there's population studies, and here in Europe, the EPIC trial, the EPIC study is really indeed one of the most important collections of data that is continuing to give us insights on how dietary patterns associate with outcomes, including cancer. So moving to how things work, in looking at this program, I think these are the things that we are going to touch on and perhaps more. I'm going to talk about angiogenesis in just one moment, but we can alter cancer cell and host metabolism. We can enhance and alter and control, modulate the immune response. I'm not going to talk about that, but I'm also involved with looking at dietary control of immunity. There's also influence on DNA repair, influence on the microbiome, which I'm also not going to have time to talk about. I'll touch on briefly. So there's many mechanisms of how diet can actually work, and these mechanisms communicate with one another. So the microbiome, the diet goes into the gut, it connects with the immune system that lines the gut. The microbiome or the microbiota communicate with the immune system communicates with other parts of the body. So this is a systems biology enterprise that we are entering, and not a reductionist one. And I know there are some journalists here who have been very enthusiastic in this topic, and one of the challenges, I think, in communicating this field to the public is not to oversimplify, and yet still give people something that they can actually understand. There's something that they can do on a pragmatic level. So I gave a TED talk on angiogenesis that was called "Can We Eat to Starve Cancer?" introducing the idea that what has been accomplished in biotechnology could be applied to study diet. In other words, the methodologies, the research tools could be converted from looking at drug development, and you can crank the turret and refocus the lens on food and some of the remarkable things that we've discovered over the years. This has been quite a popular TED Talk. Some of you I know have seen it. The last count is 11 million views, and so there's a big appetite for the topic. And what I was describing, and I'm going to update you now on what we're doing, is really beginning to take a look at whole foods that one would find in the market, and begin examining what Mother Nature has actually imbued the foods with through evolution. Now, most of the bioactive chemicals found in whole foods, especially plant-based foods, evolved from over the genuine evolution, really to provide protection or defense to plants and their fruits. So they either provided protection against pests as natural chemical insecticides, so many of the bioactives have that natural function for the plant, or also they help to attract insects for pollination. So they helped reproductive defense to protect the species. And so when man co-evolved with plants and began eating the plants, those natural chemicals began having a new job description, new function, which is that they were swallowed by humans, and they came in contact with human cells, and then a third job description became there, which is to interact with human tissues. And this is really what I call an undiscovered country. There is an enormous amount of information that we have yet to learn about what the natural chemicals in plants that serve the biological function in plants, what they do in the human body. So dietary anti-angiogenesis is a corollary to pharmaceutical anti-angiogenesis, and it goes even further, because at the very end I'll tell you a bit about some of the work we're doing looking at the other side of the equation. This is a list of different sources of dietary anti-angiogenic foods that we've actually found. I want to just show you how evidence has developed over time by taking a plant, the soybean, a plant-based food, and show you how this evidence is developed. So everybody has had probably an edamame in a Japanese restaurant. It's a very important, actually, foundational food for Western culture as well as Eastern culture. But this, in 1991, was the pivotal paper that came out of the University of Helsinki, including a Greek researcher, Ted Fotis, who inherited urine samples in a laboratory from a prior researcher who studied steroids in women and was looking at them in women. And so there had been, from a previous collaboration, frozen urine from a Japanese village, from villagers from a Japanese village outside of Kyoto, where the idea was to perhaps study their steroid content. And what this group did is they compared what kinds of foods that these subjects ate. There were 17 of them, and they were all farmers, men and women. They ate what they grew. It's a rural village outside of Kyoto. And then they looked in their urine and found discrete peaks that they could actually identify, one of which was genistein. And genistein is a phytoestrogen, which subsequently was discovered to be a dietary inhibitor of angiogenesis in vitro. And we've taken this on by working with the U.S. National Cancer Institute in a model in which we're studying angiogenesis in these rings, of aortic rings, that actually are lined with endothelial cells, which grow blood, which what blood vessels consist of. And you can stimulate the system to have this outgrowth of blood vessels as starburst. And when you put genistein, derived from soy, into the system, you can see a very potent inhibition of angiogenesis, similar to the way that tumors would grow blood vessels, and that anti-angiogenic drugs would stop those blood vessels from growing. This is what the effect of genistein is, and it's consistent and builds upon Ted Phosis's work. Now, subsequently, other groups, including this group in Taiwan, began looking at the molecular mechanisms of action, and they found many. They found that genistein inhibits in endothelial cells growth factors like VEGF, PDGF, urokinase, plasminogen activator, proteases like MMP2 and 9, and upregulate endogenous inhibitors of angiogenesis like angiostatin and endostatin and thrombospondin. So for those of you in the dietary world, this may be alphabet soup, but for those of you who are from the Institut Gustave Roussy, who have worked for many years in this area, you'll know that these are very highly valid and important targets. So this is the way that we look at this at the Angiogenesis Foundation, is to look at the entire spectrum of blood vessel growth and say, where do those targets hit? And in the X's is where the targets are hitting from soy, from blood vessel growth. And also in the box on the far right in red is where the natural inhibitors are coming up. So this is actually shifting the microenvironment towards an anti-angiogenic outcome. Now, is there any human evidence or patient translation of this data, any association? Because what you do in the lab often fails to bear fruit in people. And so this is where you jump to epidemiology. And I'm going to beg forgiveness from my colleagues associated with WHO, because I'm not going to be able to speak with the same authority about epidemiological studies. But this is a publication from 2009 examining in Asian American women childhood, adolescent, or adult soy intake, and finding that, in fact, soy intake early in life is associated with decreased incidence of breast cancer, decreased risk. And this is from 1,563 patients, so it's actually quite a large study. Now, it's a risk, and it's an association of decreased risk. What if you actually have breast cancer? So this is a huge study, 5,000 patient study, this Shanghai Breast Cancer Survival Study, in which they looked at women in China who have breast cancer and what their association is between soy intake and cancer outcome. And it showed that women who actually ate soy had up to a 29% reduction in the risk of death and a reduction in recurrence by 32%, with the association of about 10 grams of soy per day. So how much is 10 grams of soy? It's about a cup of soy milk, or 1 cup of edamame, which you can have in a restaurant anywhere, or tempeh, which is Indonesian, or soybeans. So not really unreasonable amounts is where this association comes from. Now, I just told you Asian American women, and also for risk, and also a study in Shanghai. So is this effect somehow genotypically in the Asian population, which is a very different environment? Well, this is a study in North America looking at the breast cancer family registry involving many different ethnic types, looking at the same thing. This is 6,842 patients, and finding that in an ethnically diverse cohort, the higher intake of soy reduced all-cause mortality, including cancer. So this includes Caucasians, Hispanics, African Americans, and also Asians. So we've actually been looking at this type of string of evidence, from molecular to animal, to cellular, to animal, to public health, and beginning to see, can we actually do comparative activity? And you can see soy we've studied here, and we actually compare the activity in the lab to chemotherapy and other targeted cancer drugs, and you can see it's a very interesting way to look at the opportunity to look at food activity. Green tea is another area that I think you may be familiar with, many catechins in green tea. I'm showing you this really not to just keep on talking about another food, but to show you the methodology. This is a fertilized chicken egg growing in a dish, also used to study angiogenesis, and if you can actually put onto the vascular growing network tea or any other food substance, and you can see on the right that when there's an anti-angiogenic substance in the natural compound, the blood vessels are actually dissipate. And you can also use an implant in the cornea. Here in a rabbit, this is a mouse, where you can give mice tea in drinking water and implant a substance that will grow blood vessels around the ring of the cornea. You can see there, they're growing up towards the implant, which acts like a tumor. And then if you just add drinking water, you can then quantify the effect from dietary intake of tea, and you can see much fewer blood vessels there. This can be counted, you can do the histology, you can look at the mechanisms of action. We can then actually then take it further to look at the differences between different tea types, and we looked at the differential activity between jasmine tea, sencha tea, Earl Grey tea, and then some blending. And what we found was really interesting in an in vitro assay, which is that combining the teas could actually lead to some synergy. So one tea plus one tea led to even a greater effect than the summation of them alone. So again, is there any human correlates? This is actually a study involving the National Cancer Institute in the US, prospective cohort study examining green tea and colon cancer risk. Again, an association, but this is now in 69,710 women. This is a huge study. And what they found is drinking on average 2 to 3 cups of tea a day reduced the risk associatively by 57%, which is huge. So one question that we're beginning to ask is, well, how would you actually manipulate the food? So many of you will go to a restaurant and you'll order tea and they'll give you a tea bag. Well, how do you actually get the catechins from the tea? If you just drop the tea bag and let it to settle to the bottom, there will be the catechins will dissolve out. And we wanted to quantify that, so when you actually shake the tea bag, you kind of dunk the tea bag, as some people do, you get more catechins coming out. So manipulating the food and how it's actually handled can actually impact how much of the bioactive is actually consumed. So this is really some interesting ideas. Now, the other thing about tea is that tea influences the microbiota. So the tea we drink also influences the gut, and the bacteria in the gut helps the gut change its gene activity, which then can systemically influence the vasculature as well. So again, this interconnectivity is very complicated, and we're just beginning to be able to study how these go. So I've shown you many different foods. I can't talk about them all, but I'll just finish by talking about an area that we're working on now called the Health Expedition. This is really a new area for us, a new initiative, a global initiative, and very interested in developing collaborations where we're looking at the great expeditions of the 19th and 20th century in the Amazon and the jungles and mountains and in the South Pole. And we think that the big expedition now in the 21st century is inside our body in health defense and looking at how what we do in our lives, including diet, affects that. So we're beginning to look at things like differential activity among wines, so same terroir, same grower, different grapes, and to see in the same vintage what the activity of anti-angiogenesis is. So you can see in one research we found 3 types of grapes that actually generate the most anti-angiogenic activity. We also looked at tomatoes. Tomatoes have lycopene and other carotenoids, including beta-cryptozanthin and others, that we know can influence prostate cancer. Are all tomatoes the same? No. Actually, we found that the San Marzano tomato has the highest potency, and San Marzano comes from the volcanic hills of Italy. The soil might influence it, the genetics of the plant may influence it, so we're interested in working with the agricultural world, particularly in Europe, where there's historically been great diversity of plant-based foods, and the pressures to try to do what we've done in America, which is to try to make all the foods the same, is a problem because we haven't yet fully understood how the different species are. This is the fall. We're in September in France. You go to the market in Paris, you'll see many different choices of apples. Which one might you take for cooking? Well, I leave it to the chefs. For those of us here, which apple a day might keep the doctor away for cancer? Well, we've looked at this for angiogenesis, and you can see if you look at comparative activity, the Renata apple, or Renate in French, is one of the most potent, but so is the Granny Smith and Red Delicious. And so choosing the varietal can actually have an influence. We've also begun studying in our health expedition the anatomy of a plant. So if you go to buy a frozen bag of broccoli, you'll see it's mostly the florets, and many people who buy broccoli from the market will chop off the stem and cook and steam or saute the florets, which are delicious. we wanted to ask whether or not the florets and the stem had differential activity. And when we did this, we found, in fact, that the potency of the broccoli stem had twice the amount of anti-angiogenic activity as the florets themselves. We also found this with carrots, that the green tops of the carrot was twice as potent as the orange taproot. Now, just to close, this is, how do we measure this? We are really interested in taking the tools of molecular and precision medicine that are being used in oncology today to guide our research on the connections between dietary health and cancer outcomes. So we can take a tumor, we can subject it to full genotyping, transcriptome, proteomics. We can begin to take a look at, match that with artificial intelligence to look at FDA-approved drugs or clinical trials. We think in the future dietary factors. And then there's even liquid biopsies which involve blood over time, easily obtained, to begin looking at circulating tumor DNA, exosomes, immune markers, and many other as well. So the unanswered questions that lay ahead in terms of dose and scheduling, biomarkers, augmentation of medical pharmacological treatment, and then how do you actually maintain balance, because we don't want to impair angiogenesis altogether. We want to keep normal balance, and I don't have time to show you, but there are other foods that can recruit angiogenic stem cells from the bone marrow to affect wound healing as well. So I'll just close with this quote from the Nobel laureate Albert Szent-Györgyi, who once said that discovery consists of seeing what everybody has seen and thinking what no one has thought. And I think what we're gathered here to do today is to invite, as colleagues, each other to view diet as something that everybody knows and presenting some data that might be new to each one. So I hope I've given you the beginning of some new information. Thank you.
The IGF-1 role in aging was discovered by Cynthia Kenyon and Gary Ruvkun, and we discovered the thoracic kinase, and then we also discovered what we call the sugar signaling pathway, right? RAS, PKA, controlling, and both of them. So if you have a lot of proteins, if you have a lot of sugar, all these pathways are activated, and they make the cell grow, but at the same time makes all kinds of cells in the human body grow, but at the same time they make them weaker, right? So they make them weaker and age more quickly. And so I started thinking 15 years ago, this is really a great opportunity. Why? Because a lot of these genes that I'm showing you, these proteins that I'm showing you, are oncogenes or oncoproteins or proto-oncogenes, meaning they're the normal form of what gets mutated and it becomes a cancer-causing or cancer-promoting gene, right? So then it occurred to me that by starving cells, normal cells will all respond to the starvation signal by shutting down this pathway. So you remove the protein, you remove the sugar, these pathways are downregulated in normal cells. But by definition, the cancer cells, because they have these oncogenes, let's say in the RAS gene or even in the IGF-1 receptor, they're always stuck in the on mode. So this was really a great opportunity to distinguish all normal cells from all cancer cells. And so you starve, and we knew then later on we figured out, in fact, by starving a mouse in this case, but also something very similar happens for people, the IGF-1 is lowered by 70%, so one of these key signaling pathways is really shut down, also because the inhibitor of IGF-1 signaling called IGF-BP1 is up 10 to 20-fold, right? So you have IGF-1 being lowered, IGF-BP1, its inhibitor being going much higher, and also glucose being lowered. I mean, in mice, there is a drastic reduction of glucose. In humans, not as drastic, but there is a major decrease in glucose in people that are fasting. So then we started with simple experiments, both in the US and in Italy, which was, well, we have done a lot of work in yeast, which I'm not going to show to you. But the simple experiment was just, if you take a mouse and you starve it, can you protect it by, in fact, lowering IGF-1 and lowering TOR signaling, etc., and lowering glucose. And the answer is yes. In fact, you see a major protection. This is protection against cisplatin and doxorubicin chemotherapy drugs. And on the left, you see the red line here are mice that were starved before for 48 to 60 hours before giving cisplatin. And here are the same thing, red line, and the gray line is instead the mice that received the normal food before chemotherapy. And so you don't see this very often. For example, what you see there with doxorubicin, they're all alive, all dead, based on whether they were starved or they were fed normally. Now let's see if this works. Okay, so then on the left, the mice that were starved before chemotherapy, and on the right are the ones that ate normally, and so you see that the mice starved before chemo, they're moving around like they didn't receive chemo at all, and the ones on the right, actually about 65% of them will be dead in 10 days or so. There is a drastic difference. And I'll show you some clinical data in a second. So when we first went to oncologists over 10 years ago with this, and actually David Quinn is here, he was one of the first guys that we bothered, and they said, well, show us that the fasting and the starvation does not protect the cancer cells, because even a small protection of the cancer cells will have rendered this whole thing useless. And so we went back to the lab, and there was a reason, actually, an evolutionary reason for why this shouldn't happen. And the idea is that every mutation in evolutionary biology, we know that every mutation, most mutations are deleterious, right? So cancer, because there's so many mutations, at least the hypothesis was that when you put the cancer cell in a complex environment like starvation, you really change everything, and everything is different from what it was when the cancer evolved, right? So the cancer forms slowly, and in that time, there are a lot of proteins, there are a lot of sugar, there are a lot of carbohydrates, and so the cancer now finds itself in a very different environment, and the idea, or at least the hypothesis, is that it will struggle in this environment. So starvation, so the best thing you can do for cancer cells is to keep giving the cancer whatever, the maximum nutrition, right? So the cancer benefits much more from a lot of food than the normal cells, because the normal cells have, as I just showed you, a very, they're able to switch to a starvation response mode. Anyway, so then we started to look at this. It is, in fact, a cancer cell confused in this environment. And I'm not showing you those data. Well, actually, yeah, no, there is not the fasting alone. But already fasting alone in most of the experiments that we did. So these are cycles of only about 3 days of fasting every couple of weeks or so, 1 to 2 weeks. And over we showed that in most cases, the fasting cycles alone were as good as chemotherapy. So the cancer progression was delayed as much by just giving the short cycles of fasting as it was by giving high-level chemotherapy. But I think the important thing is, and I'll show you some slides later, is that the combination seems to be much, much more powerful. Now you have a synergism, right? If you just do one or the other, as most people try to do, we always try to go the natural way or the pharmacological way, and there's two groups that really don't like to talk to each other. But really, when you combine the fasting, as you see there, with breast cancer, and this is just one cycle of fasting plus chemotherapy. In the red, you see that if the mice are fasted and given chemotherapy, now you start seeing long-term survival, and this is neuroblastoma on the right. Now you have 50% of the mice that are cancer-free long-term, but only in the combination of fasting and chemotherapy. And this is actually a study, and I just want to show you, focus on the two panels on cancer-free survival here. This is a study done by a group in Switzerland looking at lung carcinoma and mesothelioma. And here you see the control. This is cancer-free survival. So what's the percentage of mice that are cured? So of course, if you don't treat them, 0 are cured. If you give them chemotherapy, 0% are cured. If you give them fasting, 0%. And then you combine fasting with chemotherapy, and you see it goes up to 40%. and here actually 60%. And it's very similar results now we see over and over in many different tumors. So the idea is that, as shown here, normal cells, the normal organism, you starve them. There is a very coordinated response. Why? Because we have always been exposed to starvation periods, as Jean-Jacques told you earlier. Historically, we were in a very different world, and starvation was very, very common. And it is very, very common for any organism that you can think of. So normal cells are very well adapted to this. There's lowering of IGF-1, lowering of glucose. They know what to do. They increase the stress-resistant transcription factor. One of them is called FOXO. And they go into a protected, highly protected mode, anti-aging, and also anti-carcinogens. Tumor cells, instead, everything happens the same outside of the cell, but then any oncogenic mutation, which of course all cancers have, is going to lead to rebellion, essentially, the inability to respond. And these leads, of course, if you have low glucose, you have low IGF-1, and you refuse to acknowledge that this is the environment, then sensitization happens, and you struggle and you die. You know, I always say it's like going to the desert with no water and no shade, and you're running, right? So this is what cancer cells do. As long as you have water and you have shade, you could probably run in the desert. But if you now remove the shade and the water, it's just a matter of time before you're going to die, right? So, and this is the idea that we're trying to bring into the clinic. Now, something else very quickly is what we've done recently with dexamethasone. Something that we backed the clinicians to do is to remove dexamethasone for patients that didn't need it necessarily, so for whom it wasn't absolutely required, because we knew that dexamethasone increases glucose levels in the blood. And also we knew that there were data showing that the patients that have higher glucose levels respond worse and do worse during the therapy. So you see there both the use of rapamycin at the bottom and dexamethasone, which is widely used before chemotherapy. And in both cases, you see this effect on glucose, so hyperglycemia, and the same happens in patients. And there, what we just published is look at the effect of these widely used drugs and the resistance to chemotherapy. So this is chemotherapy alone, survival after chemotherapy alone, and look at survival after chemotherapy plus dexamethasone. So these are, at least in the mouse, has a devastating effect in making the chemotherapy much more toxic to the normal cells, particularly to the cardiomyocytes in the case of doxorubicin. And here is the same for rapamycin. And then here we showed that you can, just by using insulin, so this is the sensitization caused by dexamethasone, and this is just the use of insulin together with the chemotherapy. But look at what happens when you use fasting together with chemotherapy. Now you completely reverse these sensitization effects of the dexamethasone, and same thing happens for rapamycin. Just again, the importance of mechanisms, the importance of understanding how things work, because then you can go back to the clinic with a much, much clearer idea of what to do. Now, we still haven't convinced the clinicians to remove dexamethasone. They tell us, so, in many cases it's necessary, but I think, I would be very worried if I were a clinician giving dexamethasone the day before giving chemo. Okay, so then something that we recently published was what if this damage done to the cancer cells would make the cancer, make the immune system more effective in attacking the cancer. And Guido Kromer had done a number of papers looking at chemotherapy and the possibility that the chemotherapy would predispose cells to the attack by the immune system. And so we looked at this and moved to fasting mimicking diets, because most cancer patients don't want to fast, and show that here the combination of this fasting mimicking diet. This is a low protein, low carbohydrate, high fat, ketogenic-like diet, but it's also calorie restricted. severely calorie restricted. So you see here the infiltration of T cells, of immune cells in the tumor is not very high if you give them nothing. If it's not very high with the diet, it's not very high with the chemo, and now you combine the chemo and the diet, and it goes much higher. And then we actually, I'm not going to talk about the details here and the mechanisms, but certainly just to show you that by knocking down by knocking down the immune system, by inhibiting the immune system, now the FMD, the periodic fasting mimicking diet is no longer effective, meaning that it's acting, at least in this breast cancer model, mouse model, is acting by activating, the FMD is slowing down cancer progression by activating the T-cell dependent attack of cancer cells. And this is just a mechanism where then we figured out some of the genes that are involved. Is heme oxygenase 1, which is a mitochondrial enzyme that breaks down heme. It seems to be the lowering of this heme oxygenase seems to be responsible for the sensitization effect and for the opening up the cancer cell to the immune system. Okay, so this is the paper the Guido published at the same time with ours. And I don't know if he's going to talk about it, but Guido had focused on cancer mimicking, and fasting mimicking drugs instead of fasting mimicking diets. Okay, so now in the last 3 or 4 years, we've been focusing on what's next. So the starvation and the fasting mimicking diet now are causing this very wide effect. And now in collaboration with Alessio Nencioni, we now start combining it with kinase inhibitors. And you see here in a lot of scenarios with lung cancer and breast cancer and colorectal cancer, now you can combine this kinase inhibitor with the cycle of the fasting mimicking diet. And in each case, you have additive and sometimes synergistic effects. Now we're also doing it with hormone therapy. It's working very well, and all kinds of other combinations. Okay, what about clinical data? Still early. This was something that we published a long time ago, was a case study, case report of 10 patients, 6 of whom had done the same chemo, either with or without fasting, and the side effects appear to be lower, or at least they report lower side effects, going from fatigue, weakness, headaches, nausea, vomiting, etc., when they fasted together with the chemotherapy. These were cancer patients that had all kinds of cancer, so very preliminary, but very promising. Then the study that David Quinn, Tanya Dorf, and myself finished. It took us a while because patients don't like to fast, but it was finally finished and published. And again, this is a feasibility study, but showing that patients that were on 72 hours of fasting together with the chemotherapy seems to do better, or at least reported doing better, a lot better than patients that did 24 hours of fasting. This is a nice control because it tells you that against the 24 hours, it seems to be superior, of course, very small. But this is another study by University of Leiden, looking at, this is a randomized but very small study, looking at the effect of combination of fasting and chemotherapy on erythrocyte level and thrombocyte level, and again showing that the patient that fasted together with chemotherapy were protected. I mean, the blood cells were protected against the chemotherapy toxicity. This was also shown at the level of DNA. Now I hope that the work from Charité will be shown later today. And this was on 34 women, randomized crossover trial, looking at fasting mimicking diet, a low-calorie fasting mimicking diet similar to the ones that we're testing. And I think this was worked out very well, and you're going to see it later, I hope. And then we are now close, but we're maybe midway through a little bit better with multiple trials, one on breast cancer, 60 patients randomized at USC, Mayo Clinic, and I think MD Anderson now joined. There is another one on prostate, and that's almost over. Hopefully soon we'll have the results. And there is another one on prostate cancer that is fasting mimicking diet, chemotherapy, but it's in much earlier stages. And the Leiden University said 250 patients randomized on breast cancer, fasting mimicking diet called Chemo-Leave, and breast cancer drugs. And that is about halfway through. So hopefully soon enough we'll know about the results from the midpoint. So just to summarize, I used this slide that we just recently published together with a group of oncologists at Istituto Nazionale Tumori in Milan. And the idea was that we're focusing with all this precision medicine, and that's great. And I think, as I've shown, it's very powerful. But I think that the fasting mimicking diet, and potentially some ketogenic diets create a much wider net affecting glucose, insulin, ketone bodies, IGF-1. And this is just few of the hundreds and hundreds of genes and factors that are affected. So the point that we made in this cancer discovery article was that the combination is really, we think, the future. So generate a hostile environment that has differential effects, hostile for the cancer cells, not hostile for the normal cells, and then at the same time use the targeted therapy to have a very powerful effect, hopefully in a precision manner against the cancer. And so together we may start seeing, as I just showed you earlier, for example, immunotherapy. Now we're combining with immunotherapy, and the hypothesis is, can it make, maybe sometimes the immunotherapy can work on 20, 30, 40% of the patients. Can this starvation, the fasting mimicking diets, expand this and make many more patients respond to these targeted therapies? And that's all I have. Thank you very much.
Et voilà les amis, alors vous venez d'écouter la conférence de William Li et de Walter Longo. Et qui c'est qui va suivre? C'est Thomas Seyfried. À tout à l'heure les amis, au revoir.